amazing presence of Professor Alois Jungbauer. It's a very pleasure for us, Professor Alois Jungbauer, to receive you in this webinar. But before introducing what we're going to talk today, I would like to thank you on behalf of the Graduate Studies and Research Office of the Federal University of São Paulo. And also would like to thank the host for today's webinar, which is Professor Igor Bresolin and Professor Yara Bresolin. It's an honor to have those two wonderful researchers in our group. And I would like to talk a little bit more about those two amazing professionals and professors of the university. Professor Yara, she's a chemical engineer, and she graduated from the Western Paraná State University. And also, she did a doctor and master program in chemical engineering from the State University of Campinas, and also a postdoctorate from the Faculty of Pharmaceutical Sciences at the University of São Paulo, under the supervision of the Professor Adalberta Pessoa Jurno Prados, mother, since 2015. So Professor Yara, she has experienced different fields of research, but the most important is the biomolecules recovery and purification process. She used to work with the enzyme bromelain from plant sources such as pineapple and caroua, and also with polyclonal and monoclonal antibodies. In the area of biotechnology process, she has worked with extraction, precipitation, and affinity, ion exchange, and hydrophobic interaction chromatographies. She's now currently a professor at the Unifesp in the Department of Chemical Engineering, and also since 2020, she's also participated in the Parent and Science project at the Unifesp. So she's the ambassador also of this project. And also Professor Igor Bresolin. Thank you so much, Professor Igor Bresolin, for organizing this wonderful webinar. Professor Igor is also graduated in chemical engineering at the State University of Maringá. So he has a master and doctorate degrees in chemical engineering from the State University of Campinas in the area of the development of biotechnology process. So he has a postdoctorate continuous purification process at the University of Natural Resources and Life Sciences in Vienna, Austria, with these amazing professors, Alois Jungbauer. So he has experience in separation process, working in the research field of downstream processing of biomolecules, such as monoclonal, polyclonal antibodies, and enzymes, using absorption-based chromatography techniques like ion exchange and affinity. New materials such as membranes and polymeric composites have been studied and applied as alternative supports for immobilization affinity ligands. Now he's a current associated professor in the chemical engineering department at the Unifesp. So thank you so much, Professor Igor. Thank you so much, Professor Yara, and a huge thank you for Professor Jungbauer for your presence. Professor Jungbauer is from the University of Natural Resources and Life Science and Austrian Center of Industrial Biotechnology from Vienna, Austria. And Professor Igor will give the introductions for the interaction with the public. And now, Professor Igor and Professor Yara, the floor is yours. And on behalf of the Graduate Studies and Research Office, thank you so much for letting the community of Unifesp and outside from Unifesp to join this amazing opportunity to be in touch with this Professor Jungbauer. Thank you.
Thanks a lot, Camilo. Now, ladies first, Professor Yara, please.
Well, for us, it's a great pleasure to receive today in this Unifesp International Webinar Series, Professor Alois Jungbauer from University of Natural Resources and Life Science, Boku, and the Austrian Center of Industrial Biotechnology, ACB, in the wonderful city of Vienna. Professor Jungbauer was the host of me and Professor Igor in our one-year research stay in Boku the last year. Alois, thanks a lot for your acceptance to our invitation. Vielen Dank. Now, Professor Igor will present a brief CV of Professor Jungbauer.
Professor Alois Jungbauer received his PhD in food technology and biotechnology from Boku. He serves as a professor, being the head of the Institute of Bioprocess Science and Engineering at Boku. He was also honorary professor at Wenzhou University in China and visiting scientist at the Singapore Institute of Technology. He's currently working in bioengineering of proteins, plasmids, and viruses, and has developed several continuous processes for protein purification. Fundamentals of engineering in biological product recovery are the core activities of the working group led by Professor Jungbauer. This group is focused on isolation, purification, and characterization of proteins, plasmid, viruses, and cells. Currently, mass transfer and thermodynamic phenomena in bulk crystallization, chromatography, and chromatographic reactors are also being investigated. Research is also performed in the areas of stationary phase development and the generation of novel affinity ligands in affinity adsorption. Professor Jungbauer authored more than 350 publications among papers, patents, and transfer of knowledge. He has almost 10,000 citations and an H factor of 51. Currently, he is the editor-in-chief of Biotechnology Journal and editor board member of Preparative Biochemistry and Biotechnology, and also Journal of Chromatography A. Today, in this Unifesp International Webinar Series, Professor Jungbauer will present a lecture entitled Virus and Virus-like Particles and Extracellular Vesicles Purification. Just to remember to everyone who wants to ask some questions, you can perform it by the chat, either in Portuguese or English. If you prefer, you can ask in Portuguese, then we will translate to Professor Jungbauer. So, Alois is with you.
So thank you for the very warm welcome, to this webinar. Honestly, I would prefer a face-to-face meeting, but it's now in these pandemic times, it's the best what we can do out of this situation. Also, thank you those of you who write a good morning or a good afternoon in the excellent questions column. Before I start, maybe a couple of you already know me. I actually wanna introduce myself. So Yara and Igor, it was really a pleasure having you in Vienna. And when you invited me, for me it was clear that I want to share the experience what we have in this field. the problem is when you make such a seminar, you have to make kind of a hybrid between the actually a hybrid between a lecture for students, but not to bore the advanced like postdocs or the scientists. Yeah? So maybe at the very beginning, the advanced one will be a little bit bored, but it will be challenging for the students. Yeah. At the last, not challenging, it will be informative for the students, but the second part of my presentation will be more challenging for like master students or so, because I really report from our actual research. So now I want to actually present first where I'm from and actually where Yara and Igor disappeared for a year. Yeah, so last year. So this is Austria, this is Europe, Austria and Vienna in the middle of Central Europe. And actually, this is where I am from. This is in the countryside where I am born, hilly. You see also a lot of rain, similar to Brazil, because everything is green. Yeah, we have rarely such a day in. Can you hear me? Everything fine? Yeah.
Perfect. We can hear.
Yes, Professor. It's perfectly good. Yeah.
Good, good. And you see, this is actually the countryside where I'm born in the Alps. And this is Austria, known with a lot of snow and mountains. Yeah. But actually, what a lot of people do not think about Austria, at the eastern part of Austria, we have even oil fields. Yeah, so that's because we're actually the Alpine mountains end, plains towards Hungary start, and there we have also oil fields. Actually, currently, I live in Vienna, and some of you may already have been in Vienna or visited Vienna, which is a very historic town with a lot of nice buildings, mainly built in the late 19th century or earlier. So just to get a little bit the view, this is what is called the Hofburg. This was the former palace. This is our parliament. Again, part of the Hofburg, the opera house, and so on. So it's a lot. So we have also a lot of museums, yeah. And I guess Igor and Yara, do you remember when we actually visited the Dürer Museum where they have the hair, yeah? So that says a lot of interesting collections in Vienna. Also a lot of the modern art and Art Nouveau. And actually, what's quite unique for a tourist in Vienna is that you would also visit the cemetery. Usually, if you visit the town, you don't go to the cemetery. But what you see here, this is the tomb of Boltzmann, and you see the entropy is the Boltzmann constants times the logarithm of the probability. So that's here we have Mozart, Beethoven, Brahms, Schumann, and so on. A lot. Here is Schubert, and so that's and. This is actually the Freud Museum, and this is where actually the psychoanalyst Freud, Dr. Freud, actually his couch examined his patients. Yeah. So this is the university. I'm located at the Department of Biotechnology. So we have a couple of locations in our university. This is our campus, and we have a couple of departments which are really interrelated. applied genetics, biotechnology, chemistry, food science and technology, nanobiotechnology, and then the Institute of Animal Nutrition Products and Nutrition Physiology. Yeah. So we have a master PhD program, bachelor, master's PhD program in food and biotechnology, food science and technology, biotechnology, safety in the food chain, and also two interesting PhD programs. One is biomolecular technology of proteins, and the other one is bioprocess engineering. So this is bioprocess engineering. I have recently established this PhD program. And currently I am the head of the Institute of Bioprocess Science and Engineering. And we have also a pretty modern pilot plant where we can do really process development. And when we model processes, also can experimental confirm our modeling. So, but now I really want to go to the real science, yeah. And let me double check here. I have to close the presentation and then to share. Let me see, to share again. Nicht mehr teilen. Sorry. No, I sorry, maybe that was how do I have closed the presentation, and now I can share the other presentation.
Can you see on your inferior corner on your right?
Yeah, I can see.
Yeah. So just push the button.
Yeah, that's yeah. So now, actually now it should work again. Sorry for the
That's perfect. It's presenting. Thank you.
It's presenting. Great. So it's a pretty wide topic. The virus and virus-like particles and extracellular vesicles purification. These are interrelated. So it's a really wide topic. I will try to give you an overview at the beginning, and later on some of our actually research. Yeah. So an overview about virus and gene therapy, and then virus-like particles, exosomes, extracellular particles, and an overview about the analytical methods, and then do examples by the polymer grafted media and by core shell and affinity chromatography media to separate actually these molecular entities. So I think there is definitely a difference between the manufacturing of viral vaccines and viral vectors. So viral vectors, this would be for gene therapy. So that we actually modify a virus, yeah, in a way that we can introduce a gene into our body. I think this is quite interesting because we will use the natural tropism of a virus. So you know that the viruses actually do not actually systemically infect a body or so. If you have influenza, you would infect the lung. If you have other viruses like the hepatitis B, you might infect the liver and so on. So using the virus innate actually tropism or affinity to certain organs or tissues, yeah, we can direct then a recombinant virus to direct a gene to a certain actually organ. The vaccines is actually, in the viral vaccines, we want to target actually our vaccine to the immune system, yeah, to the lymphocytes, all the best. So the viral gene therapy is actually reality, yeah, is reality. So what you see here, this is the latest stage of really approved gene therapies by FDA, EMA, or the Chinese FDA. Gentizine and ocronine, these two ones are only approved in Asia or in China. The rest are approved in US and in Europe. And so what you see, they use either recombinant adenovirus, recombinant associated adeno-associated virus, herpes virus, or an oncolytic adenovirus for the transfer of the gene. And here you can see my mouse or not. And here we have the indication, usually cancer and some.
Yes, we see.
You see the mouse, yeah, great. And you have some actually diseases which actually lack a gene, yeah. So that's so there is a lot more actually in the pipeline. I will show you in the moment. So these are, but vaccines are something different, yeah. So we have like the live attenuated vaccines. Measles, mumps, rubella, rota, smallpox, chicken, yellow fever. Or this is actually live and attenuated viruses. Then we have inactivated viruses, hepatitis A, flu, polio, rabies. And then what we have, subunit or recombinant and polysaccharide also. And here come in place the hepatitis, the papillomavirus, the hepatitis B virus, and then toxoid. This is more derived from bacteria. So that you see, we have a certain commonality, but also a difference. So this is the common human viruses. So, and you see from the shape and actually the size is pretty different from Ebola virus to the parvovirus. It's very, very small virus, or the hepatitis A, or the SARS virus, or the HIV-1, the measles virus, influenza virus. But what you see, the majority of the virus, yeah, are enveloped virus. So they have an enveloped lipid bilayer, and this lipid bilayer actually is from the host cell. Yeah, so that's the, and we will come later to this problem, because then a lot of other particles which are shedded have a similar surface. So I think a lot of you may know the virus-like particles, yeah, the papilloma virus-like particles, or the hepatitis B virus-like particles. These have been quite successful actually vaccines, yeah. But we also have virus-like particles from enveloped viruses. Let me come back a little bit to the gene therapy, and then you see the majority is going on in the US, because the pharma companies can get out the most revenue of the US actually healthcare market, then Europe, Asia, yeah. And here you see the indications. A lot is cancer, monogenic diseases, infectious diseases, and so on. But cancer therapy actually is the majority. And then you see there is an increasing number of clinical trials going on, and this is still growing, and is kind of a really exploding business. And then here you see just what is actually transferred, which type of a gene are transferred to the body. So what I already mentioned, the popular viruses for the gene therapy is the adenovirus, the adeno-associated virus, alpha virus, herpes virus, some lentivirus, and then even vaccinia virus. Yeah, so that's and you see that's a completely different viruses. Like these are non-enveloped, and the others are enveloped virus. Yeah, then they could be negative or positive stranded. Yeah, and here you see the size of the virus. The adeno-associated virus is rather small, is almost in the range of a large protein, whereas let's say the herpes virus, lentivirus are much bigger. Yeah. So I think in interest of time, I don't go into the production, but if we do the downstream processing of virus and virus-like particles, yeah, we have the production, the clarification, the centrifugation, ultrafiltration, membrane adsorbers, monoliths, and so on. Yeah, we could have again ultracentrifugation or chromatography, and then again at the very end ultrafiltration, diafiltration, or also some chromatography. In some cases, we have a sterile filtration, but in other cases, sterile filtration would not work when the virus, what we have seen, is too big. Then we have to actually carry out the entire production chain under aseptic sterile conditions. So this is possible, this is doable, but is also very costly. So here are some challenges for the viral gene therapy. So the adenovirus, yeah. So we have low diffusion rates, yeah. So then we have to reduce the particle aggregation, reduce the DNA contamination, and the empty capsid separation. If we have enveloped viruses, what I have shown you before, we have low diffusion rates. They are shear sensitive, usually a very low titer, and they are very temperature sensitive. Yeah, and they are sensitive to acidic and basic conditions. So what you see below pH 5.5 and pH 8, yeah, actually, and above 8, they would be destroyed. Yeah, and the high salt concentration could also compromise stability. Yeah? So the reason is when we do like a biopharmaceutical purification, we do a virus inactivation, yeah, for antibodies at low pH, yeah, because these enveloped viruses are very sensitive to pH, and this would actually limit then our actually conditions to purify a virus. So, and what is the real challenge is we have to have a high dose. So 10 to the 11 to 10 to the 14 particles per dose. So this is enormous compared to a vaccine. So in a vaccine, we have the maximum is 10 to the 8, maybe sometimes 10 to the 9 particles per dose. Yeah, that's really the maximum in a vaccine. So you see, this is a factor of several orders of magnitudes higher. And so the scale for gene therapy is not a small scale. At the moment is a small scale because what I have shown before, all these dystrophies also, we target a small patient population. Yeah, but once we target like cancer also, the patient population may become very high, and this is then a real challenge. So here you I show you the adeno-associated virus. Yeah, usually we add 3 genes into the virus, and actually the virus is actually the vehicle which transports the gene to the target organ. Quite often it's the liver. Yeah, so that's a lot of work is done actually in the field of blood coagulation factors, factor 8, factor 9. A lot of clinical trials are underway in phase 3, but so far nothing has been approved. Yeah. these viruses are actually can be nicely produced in human embryonic kidney cells, the HEK cells. Yeah. And then what you see here, maybe if you have a closer look, we have empty and full particles. But actually, we only want to have the full particles and not the empty ones. And because the empty ones actually would block the receptors to be taken of the cells to be taken up. Yeah. So then what is called the transformation efficiency goes down. So the industrial AIV purification process is the bioreactor, cell harvest, cell lysis, verification, ultracentrifugation, ultradiafiltration, then several chromatography steps, and then ultradiafiltration. Again, what you see, a quite complex process. And here you see such a centrifuge, yeah, in industrial scale, which works at 120,000 G-forces with 40,000 RPM. And such a rotor capacity goes up to 40 liters. This one has 60 liters. There are centrifuges around which have a throughput of about 500 liters per hour. Yeah. So that's just to get an idea about the challenges of the gene therapy. So that's a non-enveloped or what is called naked virus. Yeah, but we ourselves also focus a lot on enveloped viruses and enveloped virus-like particles. So these are nanoparticles, and you see here the electron micrograph. They consist of the viral protein, do not contain the genomic material. Yeah, so this inside is empty. Outside is actually the same surface as like the host cell. The important is no safety issue, not infectious, and they do not replicate. Here we see such HIV, human immunodeficiency virus-like particles. They could be vaccine candidates, but they are candidates also for gene therapy, because later on you can actually add a gene or a DNA or an RNA to the virus-like particle. Yeah. So how do we form such enveloped VLBs? Yeah? So the non-enveloped VLBs, actually you overexpress some of these proteins, yeah, like for the papillomavirus, and in a self-assembly process, yeah, the virus-like particle is formed. Yeah. So you can do 2 layers or 3 layers. Yeah? So you can make a very complex virus-like particles. So there is a lot of room for innovation, intuition, and also ideas. Yeah. So so you just overexpress these proteins, and the particle itself forms in a self-assembly process in the cell. The same is when you actually overexpress, for instance, the HIV GAG or another membrane targeting protein, yeah, then what happens is this membrane targeting protein actually accumulates at the inner part of the surface of the cell, yeah, so at the cell membrane, yeah, and then a virus-like particle is spotted, yeah. So what you can do again, you can do a single protein, you can do 2 proteins, you can do 3 proteins. So you could even do very sophisticated ones so that the virus-like particles is tracked to the immune system, for instance, to the T cells, and also carries the antigen. Yeah. So there's a lot of actually potential around. But except the single layer virus-like particles have not yet been really exploited. The problem is actually when you overexpress such particles, then also the cell also buds microvesicles and exosomes. So the exosomes itself could be either an impurity, or the exosomes can be also a therapeutic agents. So when we actually produce virus-like particles, we don't want to have the exosomes, or we are not sure if we want to have the exosomes or not. Yeah. So here you see how these exosomes are composed of. It is also similar to a virus-like particle. So our options for envelope particles could be the CHO cells, typical Chinese hamster ovarian cells, the HEC cells, the insect cells. So we explored all three of them. Yeah. So we have done quite a lot, yeah, with the insect cells, yeah, that we co-express baculovirus, yeah, in the insect cells, yeah, and then form actually virus-like particles. Yeah. And here in this case, this would be what you see, the spike here, the influenza virus-like particles. Yeah. So this is the hemagglutinin, and here is the GAG. Right? So you see here, when we use this system, this is a high-resolution electron micrograph. This is this here, what you see is a insect cell, and how many of these particles are embodied from the insect cell. When you have a closer look, yeah, we not only see the virus-like particles in this high-resolution electron microscopy. Here you see the bar. This is 1 micrometer. Yeah. We also see the baculovirus, but we see also other virus, also extracellular vesicles. So what you already see at once is they have all the similar size, but they have different biological properties. And this is really the challenge in purification of these virus-like particles, and also is a challenge really to understand the biological system, because we are not sure that we cannot exclude that, for instance, the exosomes also trigger the immune system. And in addition, and I will show you, we have the chromatin, because we never have 100% cell viability. The cells actually will die, and they actually will release chromatin. And here you see the pearl chain here, and this is a typical actually sign appearance of chromatin in the electron micrograph. Yeah, so you see this, and then actually this is the same size as our virus-like particles. When we do some counting of particles, actually we also count them. So you see again, I think it goes too far. So we have here our HIV GAG virus-like particles. We have the macrovesicles, we have the exosomes, and they are, you see, similar in size. Then the 150 nanometers, exosomes 30 to 100 nanometers, microvesicles 100 to 350 in nanometers. So very similar in size, similar density. The virus-like particles has a density of about 1.2 gram per cubic, and the extracellular vesicles here in the range. Yeah? So there could be a possibility to separate them or not. Then we have disassembled, deformed subunit extracellulars. And this is actually our starting material. And you see here the starting material. And in this electron micrograph, you already see also this pearl strings, yeah, the chromatin. And then again, another picture of the extracellular vesicles and the virus-like particles. Here, very nice virus-like particle, and here the extracellular vesicle, the baculovirus. So that's, you see, this is a nasty mixture. So what we can do is we can do the ultracentrifugation, chromatography, membrane adsorbers, monoliths, conventional packed bed, bind elute or flow through, receptive express material, precipitation, aqueous to phase, and hybrid processes. I don't want to go in precipitation and aqueous to phase. We have not done a lot of work in this field. I have mainly in ultracentrifugation and chromatography. Here, ultracentrifugation works. Yeah. So what you see here is that's the density here. When you see this is the density gradient. And then when you fractionate, and then you can really nicely, when you see these fraction numbers, this is the GAG molecule. So this is actually the protein which forms the extracellular vesicle. And then this, here is the Western blot. Here is the STS electrophoresis. Here is the Western blot, and you see nicely that we could really separate actually from the culture supernatant the virus-like particle. So when I now go into the downstream processing, we could do, and we did quite a lot. We did the pre-processing with core beads, or what is the restricted excess material, beads, affinity chromatography, membranes, and monoliths. Yeah? What I mentioned before, the analytics is very, very challenging because there is not a single method you can use. We need transmission electron microscopy, centrifugation. We can use density gradient. We do cryo-M, dynamic light scattering. We do electrophoresis. And then we also do some mass spectrometry, ELISA, and some antibody binding. And for the separation, we use actually the conventional porous beads, the core shell, the monoliths, and the membrane adsorbers. So we have published recently this year, I think, yes, this year, a capture and purification of human immunodeficiency 1 virus-like particles, and we compared convective versus porous media. It's open access published, so you can really look into. I don't want to go into every bit and detail, so the students or those who are interested. And also the separation of virus-like particles and extracellular vesicles by flow-through and heparin affinity chromatography, also published by the end of last year. So we have our own insect cell line developed together with the Boyce-Thompson Institute to overexpress these things. Yeah. We have characterized this. We have the transcriptome and the genome data. And here you see already, I've showed you a couple of these slides. And then what we do is we go from the small scale to the large scale bioreactor. And we have shown that we can do what is called the microscale bioreactor, and the laboratory scale bioreactor gives us almost the same actual result. That when we started to do the purification, we thought is it possible to use conventional polymer grafted media for purification of virus-like particles and viruses. And then we see here, this is the adsorption isotherm, and we can bind about 1 to the 10 to the 12 or 5 times 10 to the 12 or so particles per milliliter. So we were not sure, is this possible or not? But then it's actually, we thought seeing is believing. Yeah. So because everybody thinks that this typical conventional chromatography material is not suited for virus and virus-like particle separation. So here you see the empty bead. Yeah, this is the empty bead. Then when we actually adsorb PSA, you see in this transmission electron microscopy that the bead gets dark. Yeah, and here this is actually when we zoom it out a little bit. Yeah. So that means also, because this dark stain means that the protein binds throughout the particle. Yeah? But then, actually, when we adsorb the VLP, for instance, we see that only the outer surface is stained. Here you see typically the outer surface. So, but actually, we get a much higher binding capacity, yeah, a much higher binding capacity on this spherical bead, because the beads are not completely spherical. They are kind of corrugated. That means the surface, the real surface is much higher compared to the surface what we calculate through a let's say spherical sphere. And maybe you can see it on the computer. We have developed actually a methodology to actually incorporate a fluorescent stain to the virus-like particles. And here you see such a fractal gel bead, and you see this outer halo actually here, or this outer ring, where we definitely see that the virus-like particle itself has bound on the surface of the chromatography material. So here you see what we have done: culture process, clarification, treatment, filtration, anion exchange chromatography. Yeah, and then here you see the assays, what we have done: nanoparticle tracking, HPLC, transmission electron microscopy, and then also some virus quantification to see that we remove the baculovirus. And this is how it works. So you load, and then you elute. Yeah, so purification. So this is the cell culture process, then the particle, and then the anion exchange chromatography. And what you see here, this gray zone, this is the static light scattering. At that time, we did this offline. We did. So you see the static light scattering, and with the static light scattering, then this is an indication that particles are actually separated. Because in the flow through here, you don't see any static light scattering signal, yeah, because the signal, because no particles actually pass through, everything is bound to the chromatography bead, yeah, and then we can actually separate in a couple of different actually fractions, yeah. So what you see here, that is the protein and the DNA pass through. And then when we focus on these fractions E2, E3, for instance, here we have the P24, and then we have the VP39. The VP39 is a protein from actually the baculovirus. The H1 is the protein from the influenza, and the GP64 is actually from our actually membrane-forming protein. And so what you see here, that we definitely are able to separate the baculovirus from the rest of these things. Then we do the particle quantification. So we have low particles in the flow through, and then we have a lot of particles in the main particle fraction. So what you further do is then you do an HPLC with miles or UV detection, so to discriminate actually the size and shape of the different particles. Yeah. And what we see here in E2, we have virus-like particles. In E3, we have, for instance, other particles, including exosomes. So here I show you, in interest of time, I think I'm almost done. when we do purification of particles by core shell means one is like the cup to core. We have an outer shell with a small pore, and then a porous core which binds the impurities. So all the particles go through. Yeah, the impurities are bound. Yeah. And actually you collect the virus-like particles and everything in the flow through. So actually DNA and so on, after a benzonase treatment, the DNA, everything is reduced and goes through the particles. So what you see here, the load, the flow through. So in the flow through, you have the purified material. And then we went on and separated the virus-like particles, yeah, on a heparin surface. Because it's quite interesting for a lot, let's say about 70 to 80% of all viruses, yeah, the first entry point into the cell is the binding to the heparan sulfate. Yeah? So on the cell surface, you have heparan sulfate. So heparan sulfate is considered as a viral receptor. Yeah. So, and this heparin actually mimics the surface of a cell. And so what we have here, that in the flow through, we have again the virus-like particles, and in the elution, we have the extracellular vesicles. And then you can further go on and actually measure the particle size distribution. And so what we further did is some proteome analysis, yeah, so that you see in the flow through and in the purification, we have some common proteins, yeah, but we have then also a lot of other proteins. So 74% of unique proteins interact with the HIV GAG, or which are HIV GAG virus and virus-like particles. Yeah? So that's it's quite interesting. So that we really see we have two different populations of these particles in our. And then again, you could see the envelope, the exosomes or extracellular vesicles, and the virus-like particles. Then we have the extracellular vesicles and the virus-like particles. So, in interest of time, I will conclude. So, the requirements for viral vaccines and viral gene therapy is different. So, I showed you. Yeah. So, the gene therapy is more challenging. Of course, I did not go into this detail. For the gene therapy, the revenue is much higher than for a viral vaccine. Yes, a viral vaccine has to be cheap. Yeah. In AAV, for instance, as an example, the separation, the challenge is the separation of empty and full particles. For the enveloped virus-like particles, the challenge is the separation of extracellular vesicles and chromatin, and this from the virus-like particles. And so we have several options for the purification of enveloped virus-like particles or viruses, enveloped viruses. This would be these core shell particles. There would be the conventional chromatography, and I did not go into the membrane adsorbers and the monoliths. Yeah. I think this is a very wide field, and it's really difficult actually to explain this in such a short time. But before I acknowledge my co-workers, I will actually say that I have 2 open positions for doctoral thesis in my lab, in situ removal of products by self-standing nanomembranes. Yeah. And the other one is a PhD thesis on the separation of proteins by wet-laid nanomembranes. So I want to acknowledge especially Patricia Aguilar, Katrin Reiter, Petra Steppert, all three are postdocs, yeah, who had done really superb work, yeah, that we arrived where we are now. This over is my group in Vienna, and I want to acknowledge actually our PhD program, a couple of companies, the Boyce Thompson Institute and Giorgio Carta from the University of Virginia. And according to the time schedule, I think we still have time for a couple of questions or comments. Yeah.
Yes, perfect. Thanks a lot for your marvelous lecture always. We have several questions arriving here in the chat. But the first one will be mine.
Okay, good.
Are there any coronavirus VLP being developed or purified by your working group and at Boku?
We have, I think this would be early. We made the same concept. I like the influenza virus-like particle. We made the SARS-CoV-2 virus-like particle with this spike protein. So we have it. We have it inside. Yeah, we have it in-house. Yeah. And we do currently the testing with the actually Mount Sinai Hospital in New York. Yeah, but it was too early to show you the coronavirus-like particles. Yeah, so the things. Yeah, but this is what is with, we consider this like as a platform process. Yeah. Instead of the influenza hemagglutinin, you could do any other surface protein. Yeah, but the trick is to position. How can you position this protein on the surface? Yeah, and maybe some of these virus-like particles will make it to a COVID-19 vaccine. Might be possible. Yeah.
Excellent. Other question from Natalie Del Valle. Amazing topic. I'd like to know which is the main challenge for industries to introduce continuous VLP process.
A continuous VLP process, that would be, I think, continuous VLP process, that will be the difficulties is the upstream processing, not the downstream processing. Yeah, because it has been shown when you continuously produce virus that the virus actually mutates also over time. Yeah, and then you lose productivity. For the downstream processing, I think you would apply the same principle like what you do for continuous antibody purification. The counter-current loading, yeah, you could do parallel chromatography, maybe even precipitation, all these type of things which offer continuous. Yeah, but the upstream, yeah, the bioreactor is the problem. Yeah, I don't see the questions anymore. Before I saw the questions. It's fine.
I will present it. We have another one from Professor Eliezer Ladeia Gomes. How to do a controlled delivery of vaccine substances using this presented method? How to measure the vaccine diffusion of these substances? Chromatography could be used.
What do you mean to control? What is meant with diffusion? This is what I do not understand with the question.
How to measure? Eliezer, if you can explain, put in the chat, then I will do the question again.
Yes, Igor, you can do the next question and after return to Professor Eliezer question.
Yes, but we have other question from Professor Eliezer. If molecular sieves have been tested for vaccine delivery, is it possible?
That means that molecular sieves, what means for the vaccine delivery or for the vaccine purification? This is actually what confuses me a little bit in this question.
For the vaccine delivery.
For the vaccine delivery, so that the vaccine has been incorporated in a matrix and then delivered through a molecular sieve. Yeah, is this meant?
Yes. The other one, the question from João, is given the advantages of VLP for vaccines against some diseases,
Yeah.
do you think it is possible for the pharma industry to migrate to continuous VLP vaccine production? What are the challenges regarding the pharma industry?
Yeah, what I mentioned, the continuous production actually is, you have to think about currently the baculovirus system, yeah, is working that you have the baculovirus carrying the gene, then you infect the insect cells, yeah, then you infect the insect cells, and then the virus-like particle is bodied or formed, yeah? So, or what you do, others do, like when you do the hex cells, you transfect the hex cells with a plasmid DNA, for instance, yeah? And then, actually, so you don't have a constitutive expression. And this is really because you would have to actually continuously cultivate the cells and then continuously infect the cells. So this is different to a continuous manufacturing of antibody, for instance, where you have a construct with like the CHO cells, where the antibody is actually integrated, and the gene is integrated in the genome, for instance, the gene of interest, and then produces actually constitutively the antibody. Yeah? I don't know, is this clear or not? So that's actually the formation of the virus or virus-like particles is more complex than actually the recombinant protein expression. Yeah.
Perfect. We have other questions from Professor Eliezer. How to separate the several kinds of organelles for the different applications when we talk about extracellular vesicles purification?
I think one recommendation is to use, for instance, heparin. Yeah, because if you have virus, but this will not work for all viruses, but about 70 to 80% of all enveloped viruses. Yeah, so they have affinity, yeah, to the heparin, and the other ones don't have affinity to heparin. So that's a quite interesting biological effect, yeah, to separate this. Yeah, or you use ion exchange chromatography, yeah, and do really what I showed, a linear gradient separation to have a relatively high resolution, yeah, yeah. So that could be. So there will not be one single method. Will not be a single method. I believe more on hybrid. So in the laboratory scale, definitely you would do the preparative ultracentrifugation, yeah, and then we do some chromatography. Unfortunately, there are not a lot of labs around which have access to preparative and industrial ultracentrifuge. So we worked hard to get one, yeah. So we can actually centrifuge between 1 and 50 liter of culture supernatant. So then you can concentrate, and this is a really good starting material.
Other question from Rafael Pedro Madeira da Silva Souza, our PhD student. His question is, considering that virus usually subvert to machinery for the formation of extracellular vesicles for its own production, is it possible to infer some similarity of their membrane composition? Also, when purifying VLPs by chromatography, would they be eluted together with extracellular vesicles or contaminated by the EV due to this possible similarity?
Actually, so what I showed you, the formation of extracellular vesicles, yeah? Some of them microvesicles, and the formation of exosomes and virus-like particles are kind of a different process, yeah? But they are all contain they are enveloped, yeah, by the lipid bilayer of the cell membrane. Definitely they differ in lipid composition and protein composition. But this is ongoing research where also we are involved, and which is very fascinating because it's still not fully understood. Before you are not able to separate them, it's very difficult to make a protein analysis, whatever, yeah? So this is also the reason why there is not a lot of knowledge there. But I'm pretty sure you are able, with ion exchange chromatography, some affinity chromatography, to separate these different particles. Yeah.
Next question. Professor Adalberto Pessoa Jr. Are there vaccines against COVID being developed with virus-like particle? Are there good prospects for generating the vaccine this way?
Yes, I can say yes. That's so several companies working in this field. Several companies working in the field. You're doing enveloped virus-like particles. We are a little bit stubborn, yeah, because we use the HIV gag molecule to form the virus-like particle, and the health authorities are very concerned about this, yeah, because you would actually also inject an HIV protein, yeah, but it has been shown that originally this HIV gag molecule, yeah, has been developed for an HIV vaccine, but the HIV vaccine did not work because the immune system did not recognize the gag. So we have clinical evidence. If you inject an HIV-1 gag virus-like particle, that you don't get antibodies against HIV-1. You would only get antibodies against whatever is influenza or the SARS-CoV-2 or whatever we do. Yeah. So because we expand our activities to other emerging viral diseases, yeah, West Nile virus, and the other one is Usutsu virus. I don't know if you heard about this emerging virus. For Europe, a little bit the problem because it is transmitted by migrating birds from Africa. And with the climate change, yeah, all these animals always go further north. Yeah.
We have another question from Professor Katia Ribeiro. I would like to say that the lecture was very interesting. I want to congratulate Dr. Jungbauer and his group for their work. And I'd like to ask if more and more projects should have an interdisciplinary character involving engineering, biology, and medicine, among other areas. Do you think this is important?
Definitely, definitely very important. I tell you why. Because now we started the collaboration with the immunologists. Yeah, because to understand the immunology of these particles is really important. For both, you have to understand the immunology if you want to use this virus-like particle for gene therapy. Yeah, because for gene therapy, you have to bypass the immune system. Yeah. So, like what is the AAV, the adenovirus, but also our virus-like particles. When I want to deliver the gene to the organ, yeah, it should not be actually cleared by the immune system. It's obvious, yeah, because then it would not work. On the other hand, if I use these viruses or virus-like particles for gene therapy, I want to actually get them into the immune system. That means to the lymph nodes and then the T cells, B cells, and all this as soon as possible before they are cleared from the body. Yeah. So you see, the understanding how these things behave, yeah, the immunology of these particles is extremely important. But this is not my expertise. This is a field by itself. But as long the immunologists do not have defined particles, so if they have a mixture of exosomes, microvesicles, virus-like particles, or viruses, they never can say what is the effect of the exosome, what is the effect of the virus. Yeah?
The question now is from Paulo Eduardo da Silva Cavalcante. The presentation was amazing. I would like to know if you think that steps in ultracentrifugation could damage the virus-like particles. If yes, do you think in a better step?
I think the virus-like particles are not damaged by the ultracentrifugation. I think enveloped viruses, yeah, are very sensitive to what we call the air-liquid interface. Yeah, air-liquid interface. So the forming. Yeah, so that's the problem is when you destroy the virus-like particles is if you have at the entrance or the exit of the centrifuge, when you have this industrial centrifuge, yeah, a high flow or velocity gradient. And then you know as from engineering, the high velocity gradient may actually form or induce cavitation, and this destroys the virus-like particle, but not actually the shear force, yeah, itself, yeah. And then also in ultrafiltration, not the shear force, it's the formation of foam and bubbles destroys, and not the mechanical shear. Yeah.
We have one comment from Professor Cristiane Reis Martins. the head of chemical engineering department. It's a comment regarding our stay in Vienna. With congratulations on the opportunity for always be with us here at Unifesp, and she's also very glad that two professors from chemical engineering department stayed in Jungbauer's working group with this nice area in this promising group.
Great, thank you. No, it was a pleasure. I really enjoyed your presence in Vienna. Yeah, maybe you come back for a visit. Yeah.
For sure. Also, Professor Katia Ribeiro wants to know how me and Yara collaborate to the group within our staying last year.
No, it was good. You see, we have joint publication, and there will be one more joint publication, and we with the continuous purification of the antibody, we will continue. It was a little bit interrupted or delayed everything due to this COVID-19 pandemic. Yeah. So
perfect.
Yeah. One more publication is outstanding. Yeah, so we have to do this.
Yeah, for sure.
We don't wanna.
Excellent. Any more questions? So it's the last chance. It's the last chance.
Hello, there was Everson Miranda. Yes, I met him. I think I don't know when, at which occasion was
Campinas.
it? In Campinas. Yes. Yeah.
I think there's another question here.
Yes, from Eliezer. I'm looking for a postdoctoral position related to cancer drugs delivery and molecular sieves. Is there this kind of position in Boku or in the institute? And also, thanks for your amazing lecture.
Boku not, but if you look for such a position, I would actually either check at Medical University of Vienna, Medical University of Vienna, or what is called the Max Perutz Laboratories in Vienna, because they are really working in this field, and I'm sure they if not now, but I know that they very often actually have open postdoc positions.
Yes. Alois, do you want to present any final considerations? Then we.
I think one can talk about everything, but not more than 1 hour. Yeah, so that's no. I think I really enjoyed this. I really enjoyed also your hospitality. Yeah, so that's for me it's a pleasure to present our work, and it's also our honor. And actually, this is the reason why we do these things, to reach out to other universities, to other countries, other continents. Yeah, so that's I think that's the beauty of academic life compared working in industry. Yeah, so that's also sometimes it's much harder fighting for the funding, research projects, and so on. Yeah, that's. No, thank you for this kind invitation. Yeah, and we stay in touch. Yeah.
Perfect. One last comment from Professor Everson Miranda. Congratulations for the excellent talk. And we also met in Guarujá last year during BPP.
During the BPP, yes. Yeah.
Perfect.
Good.
Well, one more time, Alois, thank you a lot for your time today with us in this international webinar series from Unifesp. It was a pleasure, and we'll be here, and also maybe for future opportunities, interchanges, to send some students from your lab.
Yeah. Good.
And Dankeschön.
Dankeschön. Bitte, gern geschehen. Yeah. So maybe next time in Baltimore at the prep conference or other occasions. Yeah, that's let's see how this works out. Yeah.
Yeah.